Hi, it's Dr. Phoenix. Today we're going to be looking at biochemical reactions. In physical science, we learned that a chemical reaction is a process that changes one set of chemicals into another. In biology, we'll see that biochemical reactions are those particular chemical reactions that occur within living things. The purpose of these chemical reactions is to help these organisms grow and develop, reproduce, and ultimately adapt to their environment so they can survive. Now, some chemical reactions occur very slowly, as in the case of iron rusting. And some chemical reactions naturally occur very fast, as in the case of hydrogen gas exploding in the presence of oxygen. Many chemical reactions, however, occur very slowly, but in the presence of what's known as a catalyst, they can occur very fast. If you recall from physical science, we did a demonstration with hydrogen peroxide. And if you recall, we said that hydrogen peroxide, H2O2, if left in light, will decompose into water and oxygen all by itself. However, this is very slow and you would not get much excitement out of watching that happen. However, in the lab, we made this occur almost instantaneously by adding a catalyst to it. And when we put a little bit of soap in the hydrogen peroxide, we saw that the reaction occurred explosively and created a huge amount of foam. We call this the elephant's toothpaste demonstration, if you recall. And so by adding a catalyst, the reaction can be made to occur much faster. If you recall, when we write down chemical reactions, there's going to be chemicals on the left side of the arrow and on the right side of the arrow. Those chemicals on the left side of the arrow are known as reactants, and on the right side, products. Somewhere over or under the arrow, we may also see enzymes written down, and those are what will result in the reaction occurring much faster. Every chemical reaction will involve an energy transformation. Energy will either be released as a product or absorbed as a reactant in order to make the reaction take place. Many of the chemical reactions that occur within living things require energy. Plants get that energy from the sun, and humans and animals get their energy from eating plants or eating animals that ate the plants. Now, chemical reactions that require energy to get them started, that energy necessary to start the chemical reaction is referred to as activation energy. So in this graph, the left side of this graph represents the reactants and the amount of energy in the reactants. The right side represents the products. You can see in order for this reaction to take place, there's a little bump right here, and that's referred to as the activation energy. So the activation energy typically in most chemical reactions is some form of heat. Heat is added in order to get the reaction started. In our bodies and in most living things, it's not feasible to add very much heat to get chemical reactions started. So enzymes are used as a biological catalyst to speed up chemical reactions. If you recall from our talk on proteins, enzymes are proteins, and they do just what a catalyst does. They speed up chemical reactions, and they do this by lowering the activation energy. If this graph represents the amount of activation energy on the blue line necessary to get a reaction started, if an enzyme was present, the amount of activation energy would be lowered. So this reaction can take place with much less activation energy needed to start it. When we look at enzymes, you're going to see quickly that they all end in ASE. The enzymes are also typically named by the reaction that it catalyzes. So in this case, maltase is the enzyme that will break down the sugar maltose. And most enzymes are named that way, so they you can pretty well figure out what they do by their name. This little animation is going to show you how an enzyme works. Here's an enzyme, maltase. It's a protein, and as proteins, it has a particular shape. There's our enzyme. Maltose is a sugar, and it is, in this case, going to be referred to as the substrate, which is the reactant, the left side of the arrow. Now notice maltase has a particular part on it that has a very specific shape, and that is called the active site. The active site is specifically designed to accept the shape of only one molecule, and in this case that one molecule will be maltose. Nothing else will fit. So as you see, maltose will fit perfectly into the active site of maltase. When it does, it forms what's known as the enzyme substrate complex. When the binding takes place, maltase will change shape ever so slightly, which will result in the bond breaking of maltose, and then will release the two glucose molecules. You can see here that maltose was simply the combination of two glucose molecules. One thing I want you to notice is maltase is unchanged in this reaction. It's still there, it was not changed, and it's not been destroyed. It is still left to catalyze more reactions. So if there's any maltose available, maltase will continue to do its job, breaking it down into glucose. Okay. Now, lactose is a similar molecule to maltose, but it's not exactly the same. You can see that it has a different shape here, and it will not fit specifically into the active site of maltase. As such, it won't bind, and maltase will not break down maltose. It is specifically going to break down maltose. Lactose will require its own enzyme, and I'm sure you can figure out the name of that enzyme from what we just discussed. The name of the enzyme that would break down lactose would be lactase. Because the shapes are so much involved in how these enzymes work, they tend to refer to how an enzyme functions as a lock and key model. Just like a lock, or just like a key fits into a particular lock and no other key will fit, same thing with the way an enzyme and a substrate fit together. Enzymes are very sensitive to the environment. You recall again that enzymes are proteins, and proteins have specific shapes. Enzymes have specific shapes. Those shapes can be affected by their environment. For instance, the temperature or the pH is very critical in terms of how enzymes function. And the enzymes in our body function best at a temperature of 98.6 degrees Fahrenheit. If that temperature goes up or down very much at all, what happens to the enzyme is it becomes denatured. It changes its shape. And not only does the entire enzyme change shape, but particularly the active site changes shape. And when that happens, the substrate will no longer fit. When an enzyme is denatured, it doesn't work anymore. In addition to temperature, pH is another very important factor in the environment. And a change of pH, becoming more basic or more acidic, can do the same thing as an increase or decrease in temperature. So our body does its very best to maintain what's known as homeostasis, or which is called a steady state. Temperature and pH, as we just discussed, our body tries to maintain a relatively steady temperature and pH. And there are many other areas where our body tries to maintain homeostasis, such as our glucose level, blood pressure, and many others. Since pH is so critical, we have in our body fluids, particularly our blood, chemicals known as buffers. A buffer, without getting into a great deal of detail right now, is a chemical that helps to keep the pH within a fluid relatively constant. Our body fluids, particularly our blood, has to be maintained between 6.5 and 7.5. And if it changes much up or down from that, those enzymes become denatured, as we just mentioned. So we have what's known as buffers in our blood to help regulate the pH. This graph represents what happens to a fluid that has a buffer in it. On the left side of the graph, on the y-axis, we have the pH of the solution, ranging from 4 to 13, and on the x-axis we have different volumes of sodium hydroxide that will be added to the solutions. This first line represents distilled water, which has no buffer in it. Please notice that the addition of just a very small amount of sodium hydroxide causes a drastic jump in the pH of distilled water. These other lines represent various concentrations of buffers added to the water, with this line being the highest concentration of buffer. Notice what happens as we're adding more and more sodium hydroxide to the pH. You can see the pH of this solution changes very little. So a buffer is a chemical that helps to keep the pH of a solution from changing very drastically, even though acid or bases are being added to it. Okay, and I think that's as much as I need you to know for now. All right, I hope that's helpful to you. Please send any questions you may have to me through my big campus, and I'll get back to you just as quick as I can. And also come to class prepared to ask questions as well. Thank you so much. Have a great day.